Hello, my name is David Sheehan. I'm Professor of Biochemistry in the Chemistry Department here at Khalifa University in Abu Dhabi. I'm also Dean of our College of Arts and Sciences. I'm a protein biochemist, and all my life I've been fascinated by the relationship between the structure and function of proteins, the key agents of biological change. Thank you for joining me for this short talk entitled COVID-19. What is a virus made of, and how can we use this knowledge against it? The talk is intended to be largely non-technical and aimed deliberately at ordinary people with no scientific background, because I know people in our community and worldwide are really challenged by this unexpected pandemic. A key message I want to give you is that knowledge is power, and scientific advances in recent years have put us in a stronger position to deal with this virus than ever before. I want to focus on the structure of the virus, the biomolecules it is made of, and how knowledge of its structure gives us a head start on its prevention and treatment. The technical name for the virus causing COVID-19 is SARS-CoV-2. This stands for Severe Acute Respiratory Syndrome, Coronavirus 2. It is a member of the coronavirus family. These are RNA viruses. This means their genetic material is ribonucleic acid, or RNA, rather than deoxyribonucleic acid, or the better known DNA. And they have protein spikes on their surfaces resembling a crown. You can see that best in the middle picture here. Corona is Latin for crown. There are at least 8 members of this family, including seasonal influenza, viruses causing the common cold, swine flu, SARS, and MERS. Coronaviruses are thought to have first evolved more than 10,000 years ago and have been responsible for several previous epidemics and pandemics in human history. In 1918, there was an outbreak of a particular kind of swine flu, which we call the Spanish flu. This caused a pandemic worldwide that killed 50 million people, and it was greatly helped by soldiers moving from Europe to other parts of the world, such as South Africa, US and Canada, and Australia. There was an outbreak in 2002-04 of SARS, which was also a pandemic. In 2009, another swine flu pandemic occurred, and in 2016, the MERS pandemic occurred, Middle East Respiratory Syndrome. In the last 20 years, therefore, much has been learned of how to manage coronaviruses. And we've learned a lot about the basic biology of these viruses. This gives us a particularly strong platform to deal with SARS-CoV-2. Coronaviruses live naturally in bird and bat populations, where they can regularly mutate or change their RNA sequence. This natural reservoir of infection is why epidemics and pandemics are recurrent. It is thought new virus variants evolve in intermediate mammalian hosts, which then can be passed on to humans. COVID-19 originated in the Chinese city of Wuhan, possibly from an illegally sold mammal called a pangolin, which is similar to an anteater. We're not 100% certain of this, but this is what most people seem to think. The Chinese government declared an epidemic in December of 2019, but it soon spread worldwide, and the World Health Organization declared a pandemic on March the 11th of 2020. So far, this pandemic has dwarfed its predecessors such as SARS and MERS, leading to the unprecedented measures that have been taken worldwide and which we are all living through now every day of the week. The virus has a large RNA molecule at its core. This is unstable and uninfective outside the virus particle. The RNA consists of almost 30,000 building blocks, and there are 4 kinds of these building blocks, and these are called ribonucleotides. They are arranged in a specific linear sequence. And when I talk about mutations or changes, what I mean is that the identity of one or other of these ribonucleotides can change. Now, I've just shown a small example here of a sequence of 12 ribonucleotides, and we know that these would encode 4 amino acids, the building blocks of proteins. So this particular sequence of RNA would encode valine, isoleucine, histidine, and serine. But if some of these letters in the RNA sequence were changed, then the sequence of the amino acids in the protein would also be changed. And this is the way these viruses mutate and vary and change all of the time. SARS-CoV-2 encodes up to 14 individual proteins. These are of 2 general kinds, replicase proteins and structural proteins. So what is the virus made of? The virus particle is very tiny. About 1,000 of them laid end to end would be equivalent to the thickness of a single human hair. All viruses consist of a genome, which is either RNA or DNA, surrounded by coat proteins. SARS-CoV-2 consists of an RNA genome, as we have seen, a surrounding biological membrane, and a layer of proteins, including characteristic coat proteins called spikes. These spikes also contain sugar molecules, so technically they are called glycoprotein, and sometimes we will say the spike glycoprotein. If we look at the life cycle of the virus, viruses have no real life of their own. They absolutely require host cells where they can make copies of themselves, and they do that by hijacking the cell's own machinery. This is what viruses want to do above all else, make millions of copies of themselves. SARS-CoV-2 specifically infects cells called pneumocytes in the human lung. This specificity comes from the binding of spike proteins to proteins of a specific receptor on the outside of the cell surface. As it happens, SARS, its nearest relative, targets the same receptor. The receptor internalizes the virus into the cell, where it is processed by the cell's own machinery, releasing the RNA molecule. Now, RNA normally in cells is translated into protein in little nanomachines called ribosomes. So the viral RNA binds to ribosomes, and the cell's protein synthesis machinery kicks in, and the cell starts to make proteins from the RNA sequence. The cell is now making the virus's own proteins. We call this process protein expression. So the viral proteins now start to be expressed. Replicase genes are expressed first, and these proteins go on to form the replicase enzyme, which is a multi-protein complex, which makes copies of the viral RNA. Later on, the ribosomes start to produce structural proteins, which go on to be assembled around the RNA, forming new virus particles. And in this way, a single virus can generate thousands of new virus particles. These can now leave the cell, killing it, and infect nearby cells, thus spreading and repeating the infection. We are just 3 months into this pandemic, but already 425 scientific papers have been published on COVID-19. Now, given the slow pace of scientific publication, which includes extensive peer review and oftentimes many revisions, this is a remarkable number of papers. Scientists all around the world are now working on this problem, We now know the virus's exact genome sequence, the three-dimensional structure of most of its proteins, something that normally takes a very long time to learn. We now know this information in great detail. Our knowledge of this virus, combined with information on its close relatives gleaned from previous pandemics, gives us a strong platform to use new science to prevent or treat this disease. So let's turn now to the questions of prevention and treatment. The simplest route to prevention is simply hand washing. The virus's lipid membrane is very sensitive to detergents, such as what we find in soaps, especially in warm water, and to high concentrations of alcohol used in hand washes, and also to chemicals used in regular cleaning agents. Because the membrane can be disrupted, this breaks up the virus. And if the virus breaks up, its RNA cannot survive outside the cell and cannot infect people, therefore. So this is not some kind of a superbug, but a very fragile structure that can only survive for a limited time outside its human host. And we can make it hard for this virus by washing our hands, by cleaning surfaces, by using alcohol hand washes. Vaccination provides the long-term route to prevention. Research into vaccination against coronaviruses has been going on for many years and has already given us the annual influenza vaccination for at-risk groups. Several companies around the world had developed candidate vaccines to other coronavirus outbreaks, but they were too late to use them in earlier pandemics. These may now provide a platform for this current pandemic. Vaccines work by exposing healthy people to all or a part of the virus, triggering an immune response. If the person later encounters the virus, antibodies are quickly produced that bind to the virus and target it for destruction. And usually the patient's not even aware of this going on. Often, vaccines are made commercially from attenuated or weakened whole viruses, passaged repeatedly in cell cultures or chicken eggs many, many times. This introduces tiny mutations or changes in the RNA sequence, which accumulate and which result over time in a less infective virus in humans. So when injected later to humans, the virus is too weak to cause any illness, but can successfully trigger immunity nonetheless. Other approaches include using killed viruses or parts of the virus, such as coat proteins. Usually, immunogenic parts of proteins and viruses are on their surface, so these are best for giving an immune response. And because we know the detailed atomic structure of the spike glycoprotein in particular, we could in principle design and produce synthetic peptides capable of triggering an immune response using this knowledge. Another promising vaccination route, which is already being followed by more than 40 companies around the world, is to attempt to insert partial RNA or DNA sequences into human cells and to express intact or fragments of the SARS-CoV-2 proteins from the body's own cells. These could train the immune system and give longer lasting immunity. This is an approach that has never been taken before. There are no vaccines on the market that have been through this kind of process before. So this is radically new science. And as I said, more than 40 companies in the world are following this line of research. Turning to treatment options. Extensive research is also going on to identify drugs for treatment of those infected with this virus. Discovering and approving a new drug from scratch normally takes many, many years. There are many layers to be gone through. There are concerns about human safety, toxicity, etc. And we have to know that a drug is safe and efficacious. However, a faster approach to drug discovery is to repurpose existing drugs, known to be both effective and safe. The World Health Organization is already trialing 4 drugs, including existing antivirals and an antimalarial. Similarly, the BCG vaccine, which was first used in 1921 to give immunity to tuberculosis and has been received by more than 3 billion people worldwide, this is undergoing study in the US, Australia, and the Netherlands, and is actually giving very promising results. It's expected the first clinical trials with the BCG vaccine for treatment of coronaviruses should be completed by the summer of 2020. Another promising approach is to use our detailed knowledge of SARS-CoV-2 proteins to identify potential drugs, using computer programs that dock the drug onto the protein. This is called in silico drug discovery because it happens inside a computer. And it's a much quicker route to drug discovery than traditional approaches. Examples of tempting targets include the spike glycoprotein and the replicase protein complex. Several companies around the world are screening libraries of thousands of already approved drugs to see if a small number of them will turn out to dock to target proteins, with a view then to repurposing them for treatment of this virus. Lastly, interferon is a protein expressed in our cells in response to viral infections. And this is a protein that can induce an antiviral state in uninfected cells nearby. They literally interfere with the virus life cycle. Now, it turns out this drug is already licensed for some human diseases, such as chronic pulmonary disorder and some cancers. So again, this is a promising, already approved drug which could be repurposed for treatment of SARS-CoV-2. We already know much about this virus and its relatives. Scientists all around the world, in academia, research centers, and industry, are focused on using our existing knowledge of the components of this virus to identify new vaccines and drug therapies, notably by repurposing existing drugs and trialing existing and new vaccines. This pandemic will not last forever, and by working together, we will eventually beat SARS-CoV-2. So please follow the advice of the health authorities and stay home. Wash your hands regularly. Practice social distancing so that by staying apart, we can stay together. Above all, stay safe. And thank you for your company today. Okay, they're sending me through some questions here. This one is, do you think the Japanese Avigin medicine can be a treatment for people with COVID-19? I'm not aware of that. You will know that there are many, many prospective medicines that we are looking to repurpose. I've mentioned 4 that are quite well known. There are many, many more. Normally, a drug treatment trial involves 3 stages of testing. You have to test the drug in healthy people. You have to test it in people with the disease, and there are very many, rules and regulations that have to be followed. Now, this takes time and it takes money, and it's quite involved. So the answer to this question is I don't know if this can be a treatment, but there are many drugs out there which I am sure will turn out to be treatments. Similarly, I'm also being asked about what they're calling the new drug, hydroxychloroquine. Now, hydroxychloroquine is most certainly not a new drug. It's been around a very long time. In fact, quinine, from which this is a synthetic derivative, has been in use since the 1600s to cure malaria. So it's a very old drug, but it's been used in many different contexts. Certainly hydroxychloroquine is being included in the World Health Organization trial. So I think there is at least some evidence that it may work, but no one has done a correct, properly designed drug testing protocol. I should point out as well, the World Health Organization are actually pushing the game on here because they're trying to develop standard rules so they can test multiple drugs in parallel under the same conditions across different countries. So they are pushing to get more quickly to a usable drug. Similarly, chloroquine is used with azithromycin. There was a study done in France, which was not a proper drug. Normally it's a double-blind kind of experiment, and it wasn't designed that way. It was simply designed to quickly see if they could get an effect in patients. The preliminary data indicated that this combination of treatments does work, and it is being taken seriously, but it has not been yet studied in a proper double-blind context. I'm asked, can COVID-19 be made? Well, COVID-19 is the disease caused by SARS-CoV-2. SARS-CoV-2 can most certainly be made in the lab. We can just design the RNA sequence. We can change nucleotides in the sequence, and we can express the proteins, and all of that's been done. And if we do that in a cell culture system, the virus will assemble as normal. So yes, it can be made synthetically. Chinese have been eating bats and other animals for a long time back. Why did this virus appear in 2020? Now, again, the answer to that question is we don't really know. I did put a question mark on the slide about the pangolin. The truth about these viruses is they mutate very rapidly. This is why it's very difficult to get vaccines against, even things like the flu. It took a very long time to get to a flu vaccine. Among viruses, these are amongst the fastest mutating viruses. I think only HIV mutates more quickly. So, and that means they change their sequence. They're always shuffling their sequence. And because they're jumping back and forth between birds and bats and other mammals, there's always the prospect of new changes, new variants. And even in the existing SARS-CoV-2 population in the world, we know there are actually two, at least, different genetic variants. So they are capable of genetically varying very, very quickly. So I guess it's just a combination of circumstances came about that enabled this virus to appear in 2020. We have often asked the same question about the Spanish flu in 1918 because that seemed to come from nowhere. It totally ravaged the world, and then it disappeared again. And as far as we know, it never came back. I'm asked about ozone treatment. I don't know about that. I'm getting lots of questions here. I'm really happy. If I go back to that question about, it says Chinese have been eating bats and other animals. I don't think it's that it came from eating bats. I think it actually, what people suspect is that it was an intermediate mammalian animal, possibly the pangolin or some other animal. We know that the live SARS-CoV-2 virus has been found, for example, in one of the big cats in New York Zoo. So these viruses do go to other mammalian species, not just humans. And so therefore, there's a kind of reservoir of infection out there all of the time. Will life ever be the same after COVID-19 regarding health precautions in daily life? I think so. And the message I'm trying to give you here in this talk is that we have many tools at our disposal. The world has, the scientific and health resources of the world are being mobilized now as never before to deal with this virus. And I think, I'm very hopeful that we will go back to normal life. These viruses have a way of appearing and then disappearing, because I think partly it's because they mutate so readily. You've got to understand as well, it's actually unusual for a viral infection to be so lethal. Normally, viral infections are quite mild because it makes no sense for a virus to kill its host. So what viruses always try to do is to make humans a little bit sick, but not to make them die. So it's actually quite unusual to have a virus that will kill you. And there are many that will kill you, but most of them don't. And in fact, among coronaviruses, most of the viruses give only mild sickness. And we know with SARS-CoV-2, more than half of people don't even get symptoms. So, it's the likely thing is that the virus will mutate again, and it'll mutate itself out of existence. And we won't hear another word about it for another few years until some other variant comes on the scene. Someone asked, do you think there's a conspiracy on this pandemic? I suppose there's always room for conspiracy theories, but I'm not really convinced there is. Even people who've worked in germ warfare will tell you that it's not regarded by the military as being a very good strategy because you can't control who it's going to hit. So I don't see what the upside was for anyone out of creating some kind of a pandemic out of thin air. You will have seen from what I said in the early part of my talk that there have been at least 3 other epidemics since in essentially the 21st century. So we've actually had 4 pandemics or epidemics in the 21st century. So I think this is not really a conspiracy. I think rather it's a function of the human activities are moving and pushing into the natural space, and we're coming into contact with animals that we normally did not come into contact in the past, and we're encountering their viruses. So that's really, I think, the change. It's the pressure that we humans are putting on natural environments out there in Africa and China and India. And because these animals have nowhere to go, we're coming very close to them, and we're going to encounter whatever diseases or sicknesses they have. What is more effective to treat patients? I'm not qualified to answer that. Why can't vaccines be developed from infected sera of an infected patient? I think that's quite possible. We actually have antibodies. You can buy them commercially now already. There are commercial antibodies to every single protein encoded by the virus. So the virus is quite immunogenic. So there's no reason to think that we won't eventually be able to develop a vaccine to it. It's just that we haven't had the leisure yet to do a proper study. Antibody assays have not been developed yet that work very well with this virus. We're still kind of developing some kind of simple assays, which will be something like the equivalent of a pregnancy kit. But over time, very soon from now, I'm sure we will have this in another few months. And the point about that is that will enable us to sort of determine who actually has had the virus and quite possibly has now recovered from the virus and is immune. Because if we have a simple test like that, then we can start to get people back to their normal lives, and we can get a much better idea of the actual epidemiology of this virus. At the moment, we're looking at death rates which are being calculated based on normally people that have come into hospital. But we've no idea how many people in the wider population are asymptomatic, and it's thought that an awful lot of people are. So when we actually do the calculation of what percentage of people that get this virus die from it, the chances are it's going to turn out to be a much smaller number than 1% or 3%, the kinds of numbers that we talk about now. because we just don't know about those people, and we haven't yet got the testing, the antibody testing, in sufficient quantity to enable us to do these studies on whole populations. The areas of research we could focus mainly during this pandemic, I think there are three. I think one is obviously vaccines, developing vaccines quickly. A second area is drug treatment, so that we can treat infected people. And the third area where I think there is a really massive need, and this pandemic has shown this need more than previous pandemics, is the testing methodologies. The test for the virus right now involves a technique called polymerase chain reaction, which is quite a laborious method for a routine test. And there's a sort of a bottleneck around this testing for that reason. The technique requires some special reagents and buffers, which it turns out were in worldwide short supply very quickly. In other words, it became a constraining factor on testing. So the PCR test is not really suitable for mass testing for millions of people. So I do think there is a need for much better testing to enable us to screen not just thousands of people, but millions and millions of people. Did the genome change while traveling from China to Europe to the Middle East? Yes, if you look at, and the great thing about knowing the sequence of the RNA virus, the RNA sequence of the virus, is that we can track minor changes in the sequence. So originally, the outbreak in China had a particular variant of this virus. About 70% of the patients had that variant. When it came to Europe, only 30% of people had that variant, and about 70% had a different variant. So the answer is yes, this virus changed, and there are various variants of this virus across the world. So it's not just one virus with just one sequence. There are slight minor changes. And this is one of the complications actually in drawing the link between the bat, the pangolin, and humans, because in fact it's not really the same sequence across those three animals. So it's hard to be conclusive that we are actually getting the same virus going from one to the other, because the virus anyway changes naturally. Okay, in terms of finding a vaccine soon, the role of the World Health Organization is key, as other stakeholders need to trust, ascertain power relations and governance to succeed. Yes, I do agree. The World Health Organization is the best bet we have to have an internationally accepted testing protocol for new vaccines. It's not big pharma. Its hands are sort of clean in this regard, and I think it will be internationally acceptable. I think if the pandemic has shown us anything, it has shown us that we really are in this together. Drawing borders around our countries won't stop a virus getting in, and you can't build a wall against this virus. We actually really need cooperation between countries, and we've seen countries scrambling to get supplies of protective equipment, ventilators, and so on. And really, it would be much better if we all collaborated together and tried to ensure that the countries that have an oversupply of ventilators can help out the other countries that don't. Where I see particular concerns coming down the road is in India and Africa. These will not have the public health resources to deal with this pandemic unless they get help from outside. So I think the World Health Organization needs to be strengthened and needs to be supported in coming up with a unified, especially testing regime for vaccines, because that would be the thing that will really put the end to this. Yeah, someone has asked me, you said the virus will mutate again. Does this mean all patients infected with it have the same virus mutation at present? And if the virus mutates so readily, why hasn't it become less lethal yet? Well, the truth is the virus does mutate, and it's not necessarily the case that every patient has exactly the same virus sequence. There are, as I said earlier, some little variants that seem to occur from time to time and from patient to patient. And this may be one reason why some people get a very serious infection and other people don't. I don't know. But it's kind of inherent in these viruses that they do mutate quite readily. Where does the virus go when a person recovers? Okay. When a person recovers, usually what that means is that their immune system has responded to the virus. It has raised antibodies to kill the virus, and any virus left in the body is broken down, because what happens is the antibodies tag on to the proteins on the surface and they trigger the immune system to destroy the virus. So it's broken down and got rid of. And any later viruses that the person encounters are broken down in the same way. And they're not even aware probably of getting the second wave. So this is why we're so concerned with looking at numbers of people who have recovered, because the working hypothesis is that they may well be immune to future reinfection. Can the virus affect pets like my cats? It seems it can. It seems it can travel into other mammals. Whether that pet will then be infective or not is really not known. This virus has only been studied really over 3 months, so it's not really, there's an awful lot we don't know. But it is certainly the case that the virus can jump from mammal to mammal, and that this is its kind of special trick. I wouldn't be surprised, I would put it that way, if it could jump across into cats. Does the heat, hot weather affect proliferation and the evolution of COVID-19? Now, if I make the analogy with the influenza virus, this virus infects northern climes, northern Europe, northern United States seasonally. Normally, it appears in maybe October, November, and it's finished by February, March. And we call this the flu season. And hospitals are geared up for this. Hospitals will increase their staffing around this time. They'll make sure they have extra equipment to deal with an influx of flu patients. And they try to have their at-risk patients vaccinated against the flu before the season comes around. So it's a very seasonal virus. And the reason it is seasonal is because people tend to go inside in the cold weather, and they're close to other people, and they're coughing and they're passing the virus around. So the virus has a playground to play in. But then when the weather gets better, people move out of doors and they're less densely packed, and the virus has less chances. So we get an element of social distancing. The virus has less chance to infect people. It's actually not known if heat and changes in climate will affect the evolution of this virus. That's the truth of it. We don't really know. My expectation would be in hot countries, in tropical countries like the one I'm speaking to you from now, my expectation would be that survival of the virus on the outside will be extremely curtailed. At the moment, the virus can last for about 3 days in a room temperature surface. But if it's outside in the open air in the hot months of the year, here in the Middle East, it probably won't survive more than a few minutes. So that may make us hopeful. But on the other hand, people will move indoors where they have air conditioning. So I think the key variable here is not so much the temperature, but that the temperature changes human behavior. And the virus then can be influenced by those changes in human behavior. But the truth is we simply do not know. Some of coronaviruses are very seasonal and some are not. Okay, if the virus mutates very fast, then what's the purpose of vaccine? Why would we need vaccination if the disease-causing agent is changing and transforming fast? Now that's a very good question. If I take again the analogy of the influenza virus, for many, many decades, people said it was insane, we'll never ever get a vaccine against influenza for that very reason. Because each season it changes and we get a different influenza every year pretty much. But it turns out that there are some common parts of the proteins on the surface of the influenza virus, which don't seem to mutate so readily. And it was really by kind of coming up with a sort of a Lego approach where you use building blocks to create a virus, trying to anticipate changes, but at the same time trying to include some common features of structure. This was what allowed companies to develop a workable influenza vaccine, which we use today. In a way, we're using the same idea by taking candidate vaccines that were developed against SARS and MERS, but were never really used on a large scale. These are our prototypes to start with, to quickly get to a vaccine for SARS-CoV-2. So I think the hope is that we can find enough commonality of structure in those structural proteins to give us a workable vaccine. I think we can foresee down the road that there will be a similar situation to the influenza virus. There will be a need for ongoing surveillance to pick up new variants and to keep tweaking the virus to keep up with its moves and give us a workable vaccine for the future. What do you think should be done in countries where social distancing cannot be done due to poor living conditions to prevent the virus? I think the most probably important thing in those countries will be to put, if possible, greater emphasis on stopping the virus getting in the first place. I think there would, we have seen examples in countries that were exposed to SARS and MERS, notably South Korea, has been incredibly successful in blocking SARS-CoV-2. Now, South Korea is a developed country, but it does have quite a dense population, but they've managed to control this very, very well by controlling travel restrictions and by contact tracing. And they haven't had to go in so much for lockdowns as other countries. So, there are examples of countries that have done a really good job of controlling this virus, not so much by social distancing and extensive lockdown, but rather by testing large numbers of the population coupled with contact tracing. So that's probably what we will end up having to do. Okay, so I've been told that my time is up now, and I just want to say again, we know a lot about this virus. Don't be frightened. Try to educate yourself. Take care of yourself. Take the best advice you're being given, and stay safe. Thank you very much for the opportunity to speak with you today.